A very good afternoon to everybody. So today we are going to study about what we have covered in lecture 5. So lecture 5, week 5's class. So week 5 class is about alternative RNA processing and editing. Okay. So what is alternative processing? Alternative processing means conversion of pre-mRNA species into more than one type of mature mRNA. That means that one mRNA can be converted into more than one kind of mature mRNA. So this alternative processing can be of many types. Especially it can be of two types. The first one is your alternative splicing and the second one is alternative poly-A processing. Okay. So just a minute. Just want to see why it's not coming. Yeah. So it should come here also. It's not coming here. Okay, let it be. So alternative splicing and alternative poly-A processing, these are the two kinds of alternating processing. That means one pre-mRNA, this is your pre-mRNA, and uh, there is some, yes. So this is your pre-mRNA, and this pre-mRNA can form more than one kind of mature mRNA. This pre-mRNA can form more than one kind of mature mRNA because the pre-mRNA can be processed in more than one ways. So if your pre-mRNA is processed in more than one way, it can lead to formation of more than one kinds of mature mRNA, correct? So this is what alternative processing is about. So now what is the advantage of this alternative processing? So basically, when your introns are spliced out, so introns uh, when introns are spliced out in different ways, it can lead to the different versions of mature mRNA. But the question here comes that introns, we are going to splice the introns, we are splicing the introns, correct? Now since we are splicing the introns, then what is the need of introns? What is the need of introns? If introns are anyways going to be spliced out, then what actually is the need of having introns? So there are needs of introns because if there are introns can only lead to alternative splicing. Second thing which is very important to understand is that introns increases the expressivity of the gene. So expressivity of a gene is 100 folds higher if the cloned fragments also contain introns. This is a very important concept, that the expressivity of the gene, it increases if you also have introns with you. So always remember that uh, these introns were earlier termed as junk DNA. But as research progressed, as we got to know more about them, we got to know that many kinds of regulatory sequences are present in introns, and these introns are not junk DNA, but these introns contain some kinds of sequences which helps in the expression of the gene, or which also helps in the differential expression of the genes. So these introns earlier, which we thought them of as junk DNA, are indeed not junk DNA, but they help in the expressivity of the genes. In fact, with the presence of these introns, the expressivity of the gene increases. It is approximately 100-fold higher increases of the expressivity of the genes. Okay, this is one thing. Now let us solve a very quick question. Average size of an exon is 150 nucleotide, 300 nucleotide, 1000 nucleotide, or 100 nucleotide. Just tell me, what is the average size of an intron? I'll give you 2 minutes to answer this question. Okay. So you have 1 more minute left. What is the average size of an exon? Tell me. Okay. So the correct answer is 150 nucleotide. So the average egg size of an exon is 150 nucleotide. Okay. Next question is, if pre-mRNA can be spliced more than one way, it leads to A. Incomplete RNAs, B. Unstable RNAs, C. RNA concatemer, and D. RNA isoforms. Okay. So I'm giving you 2 minutes. In 2 minutes, you have to answer this question. The question is, if a pre-mRNA can be spliced more than one way, it leads to incomplete RNAs, unstable RNAs, RNA concatemer, and RNA isoforms. So, tell me the answer. Right now it is 7:13. I will switch the screen after 2 minutes. Okay. 1 more minute left. Okay. Now I am switching the screen. So, question is, if pre-mRNA can be spliced more than one way, it leads to A. Incomplete RNA, B. Unstable RNA, C. RNA concatemer, and D. RNA isoforms. So, it can lead to the formation of, the correct answer is RNA isoforms. Okay. So, what is the meaning of incomplete RNA? Incomplete RNA means the RNA is not complete. That means the coding sequences which are needed to make the perfect complete RNA are incomplete. But that is not the case with incomplete RNA, because whatever the sequences are there, those sequences are already present. It's not an incomplete RNA. It has been spliced in different way. It If one pre-mRNA is spliced in more than one different ways, it just leads to the formation of a different kind of an mRNA. But that different kind of an mRNA is complete in itself. We do not say that a different kind of mRNA is not complete in itself. So therefore, this cannot be the correct answer. Second is unstable RNA. Again, why it would be unstable? Think, can it be unstable? Think. Think what it, why can it be unstable? No, because again the same concept and same logic applies over here, that how it can be unstable, because different exons are chosen to make the mature RNA, but those different exons are just creating a different kind of mature mRNA. You understand? Different kind of a mature mRNA is formed, but the mRNA is not incomplete in itself, or mRNA is not unstable in itself. RNA concatamer means it's like a chain of RNA which are all interconnected with each other. No, it's not an RNA concatamer. RNA isoform, different forms of the mRNA. Yes, by choosing different exons, we are actually creating different forms of the mRNA. So RNA isoforms is the correct answer. RNA isoform will be the correct answer. That we are actually making what we are what doing. We are doing what we are making different forms of RNA, but these are not incomplete RNA or unstable RNA or something like that. Okay. Next, we come to second lecture, which is implications of introns. So, alternative splicing means two or more polypeptide can be derived from a single gene. A single gene can give rise to two or more polypeptide by choosing of different exons, achieved by alternating the pattern of exons that are spliced together. It allows organism to carry a fewer gene in its genome, and different proteins can arise from a single mRNA. Now, this is very important to understand, that I'm just taking any arbitrary number. Okay, it doesn't mean that this is a number that actually is present inside the cell. Let's say that if we have 30,000 proteins, let's just imagine it to be 30,000 or 20,000 proteins are there in the body. I'm just telling an arbitrary number. Don't say that, ma'am, this is not the actual number and this and that. I'm just taking an arbitrary number. Okay. So now we have 30,000 proteins in our body. Now these 30,000 protein means if each gene was making one protein, that means at least 30,000 genes we must have. But do you think this is a small number or a large number? It's a large number. We cannot have these many genes, because that will create a load on the genome of the organism. It will increase the genetic content, because not only 30,000 genes are there, there are repetitive sequences, there are intronic sequences, there are other sequences which are not coding sequences. So apart from these 30,000 genomic sequences that we need, we also need those sequences also. So just imagine the amount of genetic load that the cell will have. So what we can do is, just like we say that if a person is multitasking, it reduces the number of people which are required to do a work, because same people can do multiple works. You if you suppose, if we in our homes we try to search for a maid, so we don't have a maid who can one maid who can clean, one maid who can do jhadu, other maid who can do pocha. No, we try to have a maid who can do all the works together, because that will decrease the load. So similarly, if one gene can do more than one work, and what is the work of a gene? To produce proteins. So if one gene can produce more than one proteins, it will do what? It will decrease the genetic load on the cell. So that is what happens. That is the beauty of this alternative splicing, that the genetic load, the genetic content can be less, and still it can code for that much protein which are required, because one gene can code for more than one protein. One gene can lead to the formation of more than one kind of polypeptide. And how one gene can lead to the formation of more than one kind of polypeptide? Because of the alternative splicing process. How? Because this alternative splicing, if this is your mRNA, pre-mRNA, this is your exon, intron, exon, intron, exon, intron, exon, intron, exon, intron, exon. Sometimes I am taking this, and this exons. Other time I am taking this, and this exon. Then I took this one and this one and this exon. So every time I am taking a different combination of exons, and these exons are the actual coding regions. So with by taking different combinations of exons, I am actually creating a different kind of a protein product, because now the sequence is different. And if it is a correct combination of exon, which usually happens when you're doing alternative splicing, it can lead to formation of different kinds of proteins. And these different kinds of protein means that from one genes, by just changing the pattern of exons that you are taking, you are actually making different kinds of proteins. So that's what they are saying, that different proteins can arise from a single mRNA. It is very important, and it allows the organism to carry a fewer genes in its genome. With fewer genes also, you can have what you want, and that is the beauty of this particular thing. Is it clear to everybody? I hope this is clear. Okay, then alternative splicing. Alternative splicing can occur either at specific developmental stage or different cell types. This is very important. Why this line is important? Because alternative splicing can be cell developmental stage specific or cell type specific. What I'm trying to say is that alternative splicing, it can be cell or developmental stage specific or cell type specific. Okay. So, for example, the calcitonin gene yields an mRNA that synthesizes calcitonin and that can synthesize calcitonin or calcitonin gene related peptide. Okay, and both two proteins which have total different functions. This is one example. That means one gene can synthesize two kinds of proteins. One gene can synthesize two kinds of mRNA. One is calcitonin mRNA, and the other one is calcitonin gene related peptide, which is responsible in brain. Similarly, another example is the alpha tropomycin mRNA. It can have at least eight different alternatively spliced alpha tropomyosin mRNA. This is again another example. So these are the two examples of alternative splicing. Then again, one more example, that many defects in the beta globin genes are known to exist, leading to beta thalassemias. Some of these defects are caused by mutation in the sequence of mRNA required for intron recognition, and therefore result in abnormal processing of the beta globin primary transcript. This is again another example of alternative splicing. Okay, then this is again an example of Drosophila DSCA, this CAM gene, which contain thousands of possible splice variants. This is an example. Then nuclear splicing, which involves trans-esterification reaction. This is important. We have already discussed this many number of times. Again, just for your understanding point of view, that when we talk about splicing, so splicing involves trans splicing involves two trans-esterification reactions. Okay, you understand what I'm trying to say? Okay. Then GTAG rule. What is GTAG rule? GTAG rule describes the presence of these constant dinucleotides at the first two and the last two positions of introns of nuclear genes. Splice sites are the sequences which are immediately surrounding the exon intron boundaries, and splicing junctions are recognized only in the correct pairwise combinations. This is important. GTAG rule. So GTREG rule describes the presence of this constant dinucleotide. You see, this is the intron. What they are saying that this is the intron. There is a 5 prime end of the intron from where the intron starts, and this is the 3 prime end of the introns from where the intron ends. Now, GTAG rule means that at the start of the intron there would be GT, and at the end of the intron there would be AG. This is your GTAG rule. And the splice sites are the sequences immediately surrounding the intron exon boundaries. And this is the exon and this is the intron. And your splice site is here. Splice site is here, which surrounds the intron and the exon boundaries. And splicing junctions are recognized only in correct pairwise combinations. So this is your GTAG rule, that always start of the intron would always be with GT and end of the exon would always be with AG. This is your GT and AG. Then next, now we'll come to some questions again. The first question is, introns are a common feature of A. Eukaryotes, B. Fungi, C. Prokaryotes, and D. Yeast. Hmm. Tell me the answer. 2 minutes to answer this question. 1 more minute left. Okay, let us see what is the answer. The question is, introns are a common feature of A. Eukaryotes, B. Fungi, C. Prokaryotes, and D. Yeast. So the correct answer is introns is a common feature of eukaryotes. So eukaryotes have introns. Prokaryotes do not have introns because the genome size of prokaryotes itself is very small. Okay, so we do not expect introns to be in there. The genome size itself is very small, and introns are present in eukaryotes. Eukaryotes have a lot of repetitive DNA sequences also. Again, these repetitive DNA sequences are also not much present in the case of prokaryotes. Okay. So the answer to the question is that introns are a common feature of it is eukaryotes. Okay. Next question is, um, sorry. Which of the life forms show highest rate of nucleotide synthesis? A. Humans, B. Drosophila, C. E. coli, or D. Fungi. Okay. Question is, which of the life forms show highest rate of nucleotide synthesis? Okay. Right now it is 7:33. I will switch the screen after 2 minutes. Think, 1 more minute left. Okay. Time is up. So the question is, which of the life forms show highest rate of nucleotide synthesis? And the correct answer is E. coli. So E. coli, see, E. coli in amongst this, which is the simplest life form? Humans, Drosophila, fungi, E. coli. So E. coli is the simplest life form. So the here, whichever is the simplest will have the highest rate of nucleotide synthesis. So it is your E. coli. The correct answer is E. coli has the highest rate of nucleotide synthesis. Okay. Next question is, next chapter is lecture 3, which is splicing and pathology. Okay. So here, first try to understand the four major types of introns. Four classes of introns can be distinguished on the basis of their mechanism of splicing or characteristic sequences. First is group 1 introns, group 2 introns. Group 1 introns are the self-splicing introns, and group 2 introns are also self-splicing introns. So if I'm just writing it over here, group 1 and group 2. So both group 1 and group 2 are the self-splicing introns. Then what is the difference between the two? So group 1 does not require any ATP. So this is no ATP. Group 2 also does not require ATP. Self-splicing, self-splicing. So what is the difference? They are present in fungal mitochondria, plastids, and pre-RNA of tetrahyma. And group 2 are only present in fungal mitochondria and plastids. That is the difference. Then some introns which are present in pre-mRNA, they require, are they self-splicing? No. Why they are not self-splicing? Because they require some kind of assistance from some other external enzymatic factor. And what is that another enzymatic factor that they require assistance of? That is the spliceosome. So introns which are present in pre-mRNA, okay, in pre-mRNA, so introns that are present in pre-mRNA, they require spliceosome. So they require ATP also. So they are not self-splicing. Self-splicing enzymes means which does not require any kind of enzyme or enzymatic machinery, that they can perform their function by their own. Those are your self-splicing introns. And introns which require spliceosome. Spliceosome is nothing but an enzymatic complex. So if the enzyme requires spliceosome, which is an enzymatic complex, that means that they are not this thing. They are not self-splicing introns, and therefore they require ATP also. Then introns in the pre-tRNA. Some introns are also present in the pre-tRNA, and those introns also requires ATP. Okay, so these are the four major types of introns. First is group 1 intron and group 2 introns. Group 1 and group 2 both introns are self-splicing introns. They do not require any external spliceosome complex, spliceosome machinery, or any other kind of enzymes. They can perform the function of splicing by their own. Self-splicing introns are the introns that can perform the self-splicing function by their own. They do not require any enzymatic machinery to perform the splicing function. Those introns are known as self-splicing introns. And since these introns do not require any kind of splicing machinery, any kind of special functions, therefore these introns also do not require ATP. So group 1 and group 2 introns should be clear to you. Then comes your introns which are present in pre-mRNA. They require spliceosome, therefore they require ATP as well. Introns present in pre-tRNA. Now introns which are present in pre-tRNA, they might not require spliceosome, but they require some other kind of enzymes. Okay, so enzymes are needed here also. It's not like that here enzymes are not needed. Enzymes are needed here also, but those are not the spliceosome enzymes. These enzymes are known as by different name. And yes, because they also require enzymes, so they also require ATP. They are not self-splicing introns. Introns in pre-tRNA are also not self-splicing introns. Remember this. Okay. Now, here is the steps in splicing of a pre-tRNA. See here, what are the steps in the splicing of a pre-tRNA? This is the tRNA, and this bold thing that you can see, no? Let me just draw. Yeah. This bold thing, okay. This bold thing is your intron. This is the intron of 10 to 20 nucleotides. What happens? First, there is an endonuclease. So endonuclease will cut over here. Okay. So, and over here. And this is left open like this. And this particular portion is also left open like this. And this intron is excised. Can you understand what I'm trying to say over here? This portion is removed. This portion is cut open. It forms 2,3 cyclic phosphate. And here you have an exposed OH group. Then what comes? These many things come. First is the action of endonuclease. Then these many things comes. Phosphodiesterase, kinase, lipase, and phosphatase. And when all these things comes, then you have this spliced tRNA. Ligase will join, kinase will provide the ETP, phosphate diesterase will form the phosphate diester bond, and therefore this spliced tRNA is formed. So these are the steps in the splicing of a pre-tRNA. Now, non-coding intron sequences are excised from RNA transcript in the nucleus prior to the transport to the cytoplasm. This we know that introns have to be removed before this RNA goes into the cytoplasm for the synthesis of protein. So before the translation process starts, okay, before the start of the translation process, the introns have to be removed so that whatever information is there, that is only the coding information which is present, and there is no problem for the tRNA to get confused between the coding and the non-coding transcripts. Proteins, we have to make proteins, right? So because we have to make proteins, we just need the correct sequence of the exon which has to be added over there. We don't need any other extra sequences. Therefore, before the mRNA comes to the cytoplasm, it has to be in perfect way that it can synthesize a protein. Therefore, non-coding intron sequences are excised from RNA transcripts in the nucleus prior to the transport to the cytoplasm. And then introns in tRNA precursors are removed by the concerted action of splicing endonuclease and ligase, whereas introns in some rRNA precursors are spliced out autocatalytically with no catalytic protein involved. What is this? What does this statement means? That in tRNA, when tRNA is spliced out, then you might need the external uses of some endonucleases and some ligase enzymes. But when we are talking about rRNA, then you do not need any kind of the catalytic protein from outside. It can splice out autocatalytically by itself. It can be spliced out. Okay, is it clear? Just read this point again. Okay, then we come to R loop hybridization. R loop hybridization is a technique which helped in the discovery of introns and discovery of split genes. So basically, the discovery of introns happened with the. Acha, you were reading. Okay, first you read this and then I will talk about this. Have you read it? Okay. So I hope you read this point and just gone through this image and understood that what are the steps in the splicing of pre-tRNA and how the tRNA splicing of is different from rRNA splicing. Okay. Now R loop hybridization. R loop hybridization basically was a technique that has helped in the discovery of intron. And for this, Dr. Richard Robert and Dr. Philip Sharp also got Nobel Prize because of the discovery of split genes. So it's very interesting. So we will be studying about this in subsequent lectures also. But let us first go through some questions. So this is a question which says, what is the average percentage of alternatively spliced genes in humans? A 75%, B 10%, C 65%, and D 35%. Okay, try to answer this question. Think you have 2 minutes to answer this question. Okay, right now it is 7:49. I will switch the screen after 2 minutes. Okay, see. Think. One minute left more. Think what can be the answer. Okay. So the correct answer is 75%. So the average percentage of alternatively spliced genes which is present in humans is 75%. Okay, this is the correct answer. Okay, now let us come to lecture 4, which is RNA editing. Now we are going to study about RNA editing in detail. So first we are going to study about R loop hybridization. This was the technique which has led to the discovery of what? Introns. So what is RNA loop hybridization? Let us try to understand. Pre-mRNA. See, now just try to first look at this figure so that we can understand it better. This is your DNA. Okay. And this is your mRNA. Now, this is the pre-mRNA would be of same length as DNA, but mature mRNA is much shorter than the genomic DNA. This led to the discovery of intron through R-loop hybridization. Okay, so first thing is this the concept. The concept is that when scientists discovered that This is the length of the DNA. And they have compared the length of DNA, the length of pre-mRNA, and the length of matured mRNA. And they observed that the length of DNA and length of pre-mRNA is same. Okay, length of DNA and length of pre-mRNA is same, but mature mRNA is much shorter than genomic DNA or the pre-mRNA, which means that some portion of the pre-mRNA is getting spliced out, or it is taken out of that pre-mRNA, and the mRNA which is formed, the matured mRNA, the messenger RNA, which is a matured mRNA which is formed, is of much shorter length than the DNA template from which it has been formed. If this is your DNA, okay, I'll make it here. If this is the length of the DNA and this is the length of the pre-mRNA, and this is the length of the mature mRNA. That means this some portion has been spliced out or removed from the pre-mRNA. Therefore, the mature mRNA is much shorter than its DNA template. So this was the concept that they had in their mind, and with this concept, they come to the technique of R-loop hybridization. So what they do, this is your DNA. Okay, this is the DNA and this is your RNA. So you take DNA and you take RNA, and then you do the partial denaturation of DNA under conditions that allow the RNA to displace the homologous strand. See, now what is happening? This is DNA duplex. DNA is paired with DNA, but here DNA is paired with RNA. The RNA pairs with the complementary strand of DNA, forming RNA DNA duplex, leaving a single stranded region of DNA called R loop. This technique of R loop hybridization. So the DNA which is displaced, because that particular portion of DNA has been displaced, because in that particular portion RNA has hybridized, that particular portion of DNA is known as R loop, and this technique is known as R loop hybridization. Okay. So I hope this R loop hybridization is clear. What was the concept that led to this R loop hybridization? So R loop is back as actually the displaced DNA loop. Okay, just a minute. So R loop is actually the displaced DNA loop, the displaced DNA segment. Is it clear to everybody? I hope this much is clear. Okay. So RNA editing. So up till we have studied about alternative RNA processing. Apart from alternative RNA processing, there is another way by which RNA can be processed, and this is known as RNA editing. So here sequence of the primary transcript is altered. That means here we are changing the sequence in itself. In alternative processing, were we changing the sequence? No, we were not changing the sequence. We were just what we were doing is that we were just taking the different sequence and taking different parts of the sequences and joining them together, but we were not playing with the sequence of the primary transcript. So here in RNA editing, sequence of the primary transcript is altered. This is important. Sequence, we are changing the sequence. So now RNA sequence can be changed by two ways. First is base modification, that is deamination, or insertion or deletion. That means we are modifying the uh, existing base. We are modifying the existing base by A or C deamination. Either we are modifying the existing base, or we are just deleting or adding some bases, or deleting or adding the base. Now, thus, there are two mechanisms for gene editing. So, from this, we can say that there are two mechanisms of gene editing. What are those two mechanisms of gene editing? Guide RNA mediated uridine insertion or deletion, or site specific deamination. Okay, so this is your uh, insertion and deletion of nucleotide. So, see, using a guide RNA, we can insert or delete a nucleotide. Okay, knockout or knock in we can do, and we can do the insertion and deletion of the nucleotide. Then we come to lecture 5, relevance of RNA editing. What is the relevance of RNA editing? Before this, just go through this figure so that you know what I was talking about. Just go through this figure once. Insertion and deletion, I will just increase it. Yeah, see how insertion and deletion are happening. Just 2 minutes, go through this figure and tell me if there is any problem. I hope you will have gone through it and you could understand it. Okay. Now I am switching. Let us see the next lecture. which is lecture 5, relevance of RNA editing. So RNA editing functions, these are just the examples of RNA editing functions. There is no need of going that much in detail. Just you should know what are the what are the functions of RNA editing. So AI editing in glutamate receptor, GLU are by ADR2 and CNS. Then editing of apolipo in bmRNA, RNA editing enzymes act on double stranded DNA, tRNA editing. These are all the examples. Okay. Then we come to in immunology. There are many functions of tRNA, like CU editing, for example, in ApoB, So these are the functions. Now let us come to the main part, that is the questions. Okay. So just a minute. First question is, chemical reaction that cause RNA splicing is A. Condensation, B. Hydrogen bonding, C. Coupling, and D. Trans-esterification reaction. We have studied this many a times. Okay, many a times we have studied. So tell me, what is the chemical reaction that causes the RNA splicing? Condensation, hydrogen bonding, coupling, or trans-esterification reaction? You have 3 minutes. Right now it is 8:03. I will switch the screen after 8:06. Okay, think. One more minute left. Okay, now I'm switching the screen. I mean, telling the answer. The question is chemical reaction that causes RNA splicing is condensation, hydrogen bonding, coupling, and transesterification reaction. So the correct answer is transesterification. We have already talked about that splicing requires two subsequent transesterification reactions. So I can say that two transesterification, it removes intron as a lariat structure. Okay, fine. Next question is, R loop hybridization paved way for detection of gene isoforms. True or false? Think. 2 more minutes left. Think. 1 more minute left. Okay. So the correct answer is false. R loop hybridization paved way for detecting gene isoforms. R loop hybridization has led to the discovery of introns. Okay, not gene isoforms. After later on, we can say, but it is false. Then only one base in an RNA can undergo editing. True or false? Only one base in an RNA can undergo editing. This is your question. Tell me the answer. True or false? You have 3 minutes. Right now it is 8:11. I will switch after 3 minutes. Okay? Think. Think what can be the answer. One more minute left. Okay, now I am switching the screen. Okay. So, only one base in an RNA can undergo editing. The answer is false. No, it is not that RNA one base can undergo editing. It depends. Your adenosine, your cytosine, wherever there is an amine group that can undergo deamination process. So, deamination process can be done, and then you can have insertion and deletion also. So, it's not just one base, it can be more than one base. So, the correct answer would be false. Then next question is mRNA alternate splicing always brings isoforms, true or false? So we know what is alternate splicing. Alternate splicing means that one mRNA can lead to more than one kind of polypeptides. From one single pre-mRNA, we can have more than one kind of polypeptide formation. So if we talk about this, then what can it bring more than one isoforms or not? Two more minutes left. One more minute left. Okay. So the correct answer is true. Obviously, how it can be true? Think, because when we are doing the alternative splicing, what we are doing, when we are doing alternative splicing, we are making more than one kind of polypeptide of the same mRNA. And when we are making more than one kind of polypeptide of the same mRNA, what we are doing, we are actually bringing the isoform. So the correct answer is true. Next question is, prokaryotic RNA polymerase has only two subunits. True or false? Prokaryotic RNA polymerase has only two subunits. Is it true? 2 more minutes left. Okay. So prokaryotic RNA polymerase has only two subunits. It is a false. Why? What are the subunits of prokaryotic RNA polymerase? Alpha, alpha, alpha. What has happened? Alpha 2, alpha, alpha, beta, beta dash, omega, sigma. These are the different subunits of RNA polymerase. So it is false. Okay. So we have completed the questions. So in this class, what we have studied, so we have studied about alternative RNA processing and editing. This was the class. In this, we have studied about what is alternative processing. Alternative processing is the conversion of pre-mRNA species into more than one type of mature mRNA. That is your alternative RNA processing. This alternative RNA processing can occur by two ways. First is alternative splicing, and second one is alternative poly A processing. Alternative splicing means that one mRNA is spliced in more than one kind of way. Alternative poly A processing means that there are many poly A sites which are present in the pre-mRNA, and we can choose which poly A site we have to choose each and every time, and that will make differences. So alternative splicing and alternative poly A processing. Then intron advantage. What are the advantages of intron? Because it helps in alternative splicing, and expressivity of the gene is 100 folds higher if the clone fragment is also contained in introns. Then we studied about alternative processing, the advantages of alternative processing. That alternative processing allows the organism to carry fewer genes in its genome. It helps in making different proteins from a single mRNA. So these are the advantages of alternative processing. Then alternative splicing has many examples in real life. Some of the examples that we have studied, that it can occur at specific developmental stages, and for example calcitonin and calcitonin gene related peptide, or alternative splicing has other examples like beta thalassemia and all this. So these are the examples of alternative splicing. Then the splicing requires two trans esterification reaction. This we have also studied in our previous classes. Here also we are studying that two subsequent trans esterification reactions are required in splicing. Then we have studied about the GTAG rule. What is GTAG rule? That at the start of the intron has to be GT and the end of the intron has to be AG. This is your GTAG rule. Then what are the four different types of introns? Group 1 introns, group 2 introns, introns in pre-mRNA, introns in pre-tRNA. Group 1 introns and group 2 introns are self-splicing introns, which does not require ATP. Introns which are present in pre-mRNA, they require spliceosome as the machinery to splice the introns, and hence they require ATP. Introns which are present in tRNA, they do not require as such spliceosome, but still they require some other kind of enzymes, and because they require some other kinds of enzymes, they require ATP. Then what are the steps in splicing of a pre-tRNA? We have studied that there are what are the different steps in the splicing of pre-tRNA. You can see that first there is an endonuclease attack. When the endonuclease attack happen, then the intron is excised and you have two open ends. First is at the OH end and second creates a phosphate end, which is actually a cyclic phosphate that has been created. 2,3 cyclic phosphate. So first your enzyme which is acting is endonuclease. And what happens that after the intron has been excised, it is acted upon by phosphodiesterase, kinase, ligase, and phosphatase. And then the open ends are so complete splicing of the introns happens then. Then we have studied that how the splicing of tRNA is different from splicing of rRNA. In rRNA, precursor are spliced out autocatalytically. Okay, autocatalytic splicing of precursors happens in the case of rRNA. Then we have studied about R-loop hybridization. What is R-loop hybridization? What is the concept of R-loop hybridization? That the length of the mature mRNA is much more shortened than that of DNA. That is the concept of R-loop hybridization. And then the R-loop is basically the displaced DNA fragment that comes. That is your R-loop. Then RNA editing. We can also change the sequence of the primary transcript, and there are two types of modifications: base modification, insertion, and deletion. And it can be achieved through guide RNA mediated urine insertion, deletion, or site specific deamination. And then we have studied the various functions of RNA editing. There are various functions of RNA editing that can be studied. So, all this we have studied, and this all we have studied about lecture 5. So I just want you to go through this figure and understand that how. I'll just do it like this. Just go through this figure and try to understand this, that how it happens. Go through this figure and see it. See, here it is written UCAG, and here it is written UCAG, UCAG, UCAG. So how it happens? UUU. UUC. So, how are you reading it? That is what is important. This is the first letter, this is the second letter, this is the third letter. So, U, then UUC, then UUA, then UUG. Here comes UCU, UCC, UCA, UCG. So, this is the first letter, this is second letter, this is third letter. ये वाले बॉक्स में first and third, first, second, and third, first, second, and third. Change the third in this. All the third letters are same in this box. All the third letters are same in this box. You are no, no. All the first and the second letters are same in this box. Third letter is changing here. First and second are same. Third is changing. First and second are same. Third is changing. See here. Let us take any one of this. Yes. First and second are A only. Then UCAG. UCAG. GAGA, UCAG. So just 2 minutes read this so that you can understand how the genetic code is read. See here. Could you understand this? Okay. I hope you understood this. So, this is the way how you have to read it. U U C So, like this, you read it. This was a thing that I wanted to explain you, and I think I just left it so I couldn't understand. I make you understand. So, I hope this is clear to everybody. If you have any questions, you can ask me. Otherwise, we'll just complete the class now. Do you have any questions? Does anybody has any questions? If anybody has any question, just ask me. Otherwise, I'll just switch off this thing. I mean, end the class. Clear to everybody? Everything? Any doubts on this? Any question that you could not understand? Tell me. This transesterification concept, I hope this is clear to everybody. Okay, then. Then we'll meet in the next class, in class lecture 6. Okay. Bye, everybody. I'll leave it here then.